DNA replication is going to begin at a site called the origin of replication. Where does it begin? At the origin of replication. That at least makes sense. The origin of replication is a location on the chromosome, so a segment of DNA that allows certain enzymes to come in and begin this process. And from that origin of replication, we're going to move in both directions. So we're going to expand, we're going to open up this double helix, we're going to unzip the genes, and then in both directions, we are going to have a replication fork, and the daughter strands of DNA are going to arise from these origins of replication. Now, in eukaryotic organisms, we have linear DNA. The entire chromosome is one long string of DNA, and this process will continue until the entire chromosome has been duplicated. Now, that would take a very long time to go from one origin of replication and then copy the entire chromosome. But fortunately, in our DNA, we have several origins of replication per chromosome. So it can start at multiple sites. And then each one of these sites, as it expands, eventually it runs into the daughter strands being created by the other sites, and they kind of combine. I'll show you an image that makes this a little bit clearer in a moment. Now, bacteria have a slightly different situation because they don't have linear DNA. They actually have circular chromosomes. All of their DNA goes in one big loop, which is interesting. And they also don't have a nucleus to protect it. So if you can see down here, on the left-hand side, we have a circular chromosome being replicated. You have an origin of replication there, and as we are creating daughter DNA molecules at this origin of replication, right, so each strand is being pulled away from each other, and then this light blue segment is being added in, that's the new DNA being replicated. We are peeling away one circular chromosome and leaving behind another circular chromosome at the end. So the whole process allows us to kind of peel one segment of DNA away from the other one. You end up with two copies of that circular chromosome at the end. Now, on the other hand, on the right-hand side of this slide here, we have linear DNA, and you can see that there are multiple origins of replication. And when we open up the DNA double helix, when we separate the hydrogen bonds of the base pairs at any of these origins of replication, we create a little bubble in the line. It's no longer a perfectly straight parallel line. When we separate it, we get a little bubble there with a replication fork at either side. And then as these forks expand, as these bubbles get larger and larger, we're replicating more and more DNA, and eventually these replication bubbles run into each other, and we have the entire chromosome replicated. Having multiple origins of replication makes this process much, much quicker. Now, when would this occur? When do we do DNA replication? You might remember when we were talking about mitosis that there was a phase called interphase, right? Interphase is most of a cell's life. It's when it's not actively dividing. It's when it's going about the business of being a cell, but it's also when it duplicates its DNA. There's a phase called S phase, and the S stands for synthesis. What are we synthesizing? DNA. We are synthesizing the DNA. That's when we go from having one chromatid per chromosome to two chromatids per chromosome, right? Each chromatid gets a partner. Every chromosome has two legs on it at the end of S phase. DNA replication is the process that gives us those sister chromatids. So that's when this is happening. During S phase is when DNA replication is going to occur, and the ultimate product of DNA replication is that every chromosome is going to have two chromatids joined together at the centromere. Now, DNA polymerase can't do this job entirely by itself. It would be nice, but generally in a cell, you have to get a whole crew of enzymes working together in order to complete a complicated process like this. The job of DNA polymerases is just to synthesize the new DNA strand, but there's several other tasks associated with DNA replication that polymerases by themselves cannot accomplish. This animation actually shows you the entire replication process happening at the rapid speed which it occurs inside of your cells. And you can see that there's a number of different enzymes that are all working together in this kind of protein complex here. There's a dark blue one that we are feeding all of the parent strand of DNA into, right? That's right there. You can see that dark one up front, and then it's on the left-hand side when it shifts to the wide view here. That is just splitting the DNA molecule into two strings. It's unzipping the DNA molecule, breaking those hydrogen bonds, separating it into the two strands. So we got the one that's splitting it into two strands. We also got ones that, if you take a look, these light green ones here, they are taking the single strand and they are turning it back into a double helix. So those are your polymerases. They are creating the new double helix. They're creating the daughter strands of DNA. One of the difficult things about DNA replication to wrap your head around is that DNA replication can only occur in one particular direction. And because the strands are opposite to each other, you can see that the solution that these molecules have come up with is that they have to fold half of the DNA back in this hairpin loop and then replicate it in these discontinuous segments here as it goes, and then they release it and then fold another segment of DNA back and replicate that, and go on and on and on. The other DNA segment, on the other hand, can just go straight in and it can be replicated immediately. These two strands of DNA, the one that's being replicated continuously is called the leading strand, and the one that has to be folded back in these hairpin loops on a constant basis is referred to as the lagging strand, and we'll talk about each one of those by themselves. But I love this animation because this really gives you an idea of how rapidly DNA replication proceeds and how many different enzymes are involved and how complicated the process really is. Now, if you don't understand everything that's happening in this animation here, that is okay. Breathe a sigh of relief. It's fine. We're going to go through the enzymes one by one and learn their names, and we're going to learn exactly what it is that each one of them does. And then later you're going to come back to this video and you're going to see it with a little bit more understanding. Now on this next slide, we have a little bit more of a bird's eye view, a little bit better of a picture of what's going on here. So we have the origin of replication at some point in the middle of the DNA molecule, and we break the hydrogen bonds there in order to open it up. When you peel the two halves away from each other, you get a replication fork on the left and a replication fork on the right. Now, the direction of DNA replication can only occur in the 5' to 3' direction. That is, we have to add new DNA nucleotides to the 3' end of the string that we are working on. Let's say we have two DNA strands that are running anti-parallel to each other. So we have one that is going in the 5' to 3' direction. So we got the 5' end over here and the 3' end over here. What's important to know about 5' and 3' is that that's just what we call opposite ends of the same DNA strand. So we got 5' to 3'. The opposite DNA strand is going to be 3' to 5'. Now here, where I opened it up in the center, here, let me get the bases in. Now here, in the very center, where I opened up the strand to create this little replication bubble, this is the origin of replication. On either side of this replication bubble, we have what's called a replication fork. DNA polymerase is going to begin replication at this point, at the origin of replication. So it's going to create a new segment of DNA going towards this replication fork. Now, on the new segment of DNA, this will be the 5' end, and this is the 3' end. Same thing on this strand down here. We're going to add a new segment of DNA attached to the parent strand. Since this is the 3' end and this is the 5' end, on the new strand that we are synthesizing, this will be the 5' end and this will be the 3' end. Here's the hitch. DNA replication, DNA polymerase, can only add new bases to the 3' end of the molecule that it's working on. So DNA replication always proceeds in the 5' to 3' direction. 5' to 3' direction here. So from the origin of replication, moving to the left, It's easy peasy because as we keep opening up, unzipping this DNA molecule, there's more and more bases being exposed, and we can just keep cruising down here. DNA polymerase is going to ride along this DNA molecule, adding new bases in the 5' to 3' direction. Down here, as we keep opening it up, DNA polymerase is going to ride, adding new bases to the growing DNA strands that it's creating, that it's replicating, that it's synthesizing. And it's going to move in the 5' to 3' direction. However, on the opposite side of the origin of replication, well, this is still the 5' direction over here, and this is the 3', but we can only move in this direction. So we keep opening up more and more bases, but we have to essentially keep running over here and starting a new DNA segment in order to fill in the gap. I create a little bit more of a gap, I have to make a new DNA segment in order to catch up. So on one side of the origin of replication, we're going to replicate continuously. This is called the leading strand. Okay? On the other strand, on the opposite side of the origin of replication, this is the leading strand. The leading strand is the one that can just keep going in the direction it's already going. No matter how much DNA we open up, we can just keep cruising in that same direction. But on the opposite strand, we can only go in segments. Because every little segment we make is going to have to be from 5' to 3', which, as we keep opening up this replication fork, we're going to have to start towards the replication fork and then work our way backwards here. That strand is called the lagging strand. The leading strand can be replicated continuously. Whereas the lagging strand has to be replicated discontinuously. Discontinuously means that we have to do it in segments. Okay? That's all this means. You can only replicate DNA in the 5' to 3' direction, and because the strands are anti-parallel, if you have one strand is replicated continuously, the other strand has to be replicated discontinuously. It's got to be done in little segments because proteins don't have any method of solving that problem. To review all of that, we have a double helix, a two-stranded DNA molecule, two polynucleotides, two sugar phosphate backbones, and they are held together by base pairs. Those are hydrogen bonds between the A's and T's and the C's and G's. In order to replicate, we have to open it up at an origin of replication, creating this replication bubble. Each strand of DNA can only be replicated in the 5' to 3' direction. So let's ignore everything to the right-hand side here. We're going to simplify. This strand, the leading strand, is the strand for which the 5' to 3' direction takes it towards the replication fork. So it can just keep going and going and going until it reaches the end of the DNA molecule. The lagging strand is the DNA molecule that's being synthesized, which is moving in the opposite direction of where the replication fork is heading. So we can only go this direction, this direction, this direction in order to make that work. And as you might suspect, when we talk about DNA replication on the leading strand, it is very simple compared to DNA replication on the lagging strand. These little segments of DNA that have to be made on the lagging strand, they get a special name. They're called Okazaki fragments. They were discovered by a husband and wife team, both of whom were called Okazaki, so Okazaki fragments. The Okazaki fragments are the segments of DNA which are discontinuously replicated on the lagging strand. Now, eventually, these little segments, we're going to have to bind them together to make one single unified daughter strand of DNA, but that will come a little bit later.